Welcome back for the second session of today's class. I have put across these movie posters as these movies are based on the topic which I'm going to be dealing today. So the content for this particular session, we have finished genetic code. So it is going to be mutations. And the specific learning objectives for this session will be at the end of the session, you should be able to define mutations, classify point mutations based on the type of nucleotide altered, categorize point mutation based on consequence, citing examples, and explain the frame shift mutations and its consequences in protein biosynthesis. Let us define first what is mutations. Mutations results from changes in the structure of the DNA. Or one more way of defining mutation is that is they are the changes in the nucleotide sequence of the DNA. Now the changes in the DNA structure can be a small change or it could be a large change. A small change could be a single base change, that is a single nucleotide sequence change in the DNA structure, or it could be as large as the DNA strand breaks, DNA cross-linking with proteins, etc. Now, the mutations can occur both in the germ cell line as well as in the somatic cells. So, mutations in the germ cell line can be passed on to the next progeny, whereas mutations in the somatic cells does not pass on to the next generation. Therefore, mutations in the germ cell line can result in certain diseases, that is the inherited diseases, whereas mutations in the somatic cells results in cancer. Some of these mutations may be repaired, whereas others cannot be repaired, and some becomes irreversible. Coming to causes of mutations, mutations can be spontaneous or they can be induced. So spontaneous mutations occurs at a rate of about 10 to the power of minus 7 to 10 to the power of minus 6 per cell per generation. Spontaneous mutations includes deamination of the bases. For example, deamination of cytosine can result in the formation of uracil. Similarly, deamination of adenine can result in the formation of hypoxanthine. Now, this uracil and hypoxanthine, even though are normal bases, but their presence in the DNA is abnormal. There may also be loss of the entire base itself. That is, there may be a spontaneous depurination, or even there may be loss of the pyrimidine bases also. So this creates what is called as AP site, that is apyriminic site or apyrimidinic site. Now these type of mutations occur when there is a escape from the repair by the base excision repair mechanisms. Recall, under the DNA repair mechanism, you would have studied that these type of spontaneous changes which occurs in the structure of the DNA can be repaired by the base excision repair mechanism. But if there is no repair, these results in mutations. Now, the second major cause will be due to the mutations being induced by various environmental factors. For example, it could be chemicals. Okay, so chemical mutagens which can cause damage to the DNA, and many of these chemical mutagens, when they cause damage to the DNA, they can also result in cancer formation. So therefore, they're also carcinogens, but not necessary that all mutagens can result in cancer formation. Also, there may be exposure to UV radiations. So these radiations can also damage the DNA. The most common damage of UV radiations will be the formation of thymine dimers. Now again, these damages can be repaired. For example, the thymine dimers are repaired by the nucleotide excision repair mechanisms. But if it does not get repaired, then it results in the mutations. That is a permanent damage to the DNA. Ionizing radiations can cause gross damages to the DNA, that is, they can cause the single strand or double strand DNA breaks, etc. Now, apart from this, we may also have certain other endogenous factors which can also cause damage to the DNA. Recall, under the chapter of free radicals, you would have studied that various reactive oxygen species like the superoxide ion, hydroxyl radical, nitric oxide radical, etc. can cause damage to the DNA. That is, they can induce mutations. So which can also sometimes result in a very gross damages to the DNA. Now, Mutations can also occur during the normal processes which are taking place in the nucleus. For example, certain replication errors can also result in mutations. Again, these replication errors which occur are repaired by mismatch repair mechanism, but if it goes unnoticed, or if it does not get repaired, then it can result in the replication errors. So even errors can also occur during integration of transposons or during the recombination events. So ultimately, all these different causes results in permanent changes in the DNA, which sometimes may lead to cancer development, or even sometimes can, if it is in the germline line, can result in inherited disorders. Coming to the classification or types of mutations. So the small mutations which takes place in the DNA structure can be broadly classified into two types. That is, point mutations which occur due to base substitution, or the frame shift mutations which occur due to insertion or deletion of the bases. Point mutations can be further subclassified as silent mutation, missense mutation, and nonsense mutation. Whereas frame shift mutations occurs because of deletion or because of the insertion of a nucleotide base or bases. Let me explain in detail the different types of mutations. Coming to the first one, that is the point mutations. Point mutation occurs when there is a single base change in the DNA structure. That is, alteration in the single nucleotide of the gene, which may be substituted or altered. Now, the origin of these point mutations could be spontaneous, or they may be caused by errors in DNA replication, or they can also be induced by mutagens. Now, the consequence of this point mutation may go unnoticed, or sometimes it may result in gross changes in the functioning of the protein which is being coded by the gene containing this point mutations. The best example we can think of is sickle cell anemia. As I already said, point mutation occurs when there is a change in the nucleotide base in the DNA structure. That is, there is a base substitution. So in place of one particular base, if another base is present, then it is the point mutation. Now, based on the type of base which is being substituted, we have two types of mutations. The first is called as transition. Now, in the transition type of mutation, there is replacement of a purine to a purine, or also a pyrimidine to a pyrimidine. For example, adenine can be replaced with guanine, or guanine can be replaced with adenine. Similarly, cytosine is being replaced with thymine, and thymine is being replaced with cytosine. So this type of mutations are called as transition. That is, where a purine is being replaced by purine only, and similarly pyrimidine is being replaced by pyrimidine only. Whereas the second type of mutation based on this is the transversion, where there is a replacement of a purine with a pyrimidine and vice versa. For example, thymine can be replaced with the adenine or guanine. That is, the thymine is the pyrimidine here being replaced with the purine, either adenine or guanine. And in a similar fashion, you can see the other bases also which are being replaced. So here it is a purine is being replaced by pyrimidine and the other way around. Mutations can also be classified, that is, point mutations can be also classified based on the consequences or effects of this point mutation, which is also important for us to know. So based on these effects or consequences of these point mutations, mutations can be classified as silent mutations, missense mutations, and nonsense mutations. Remember, these are the subclassification of point mutations based on the effects. Whereas based on the base changes, we have the transition and transversion. Let us understand the concept of this silent, missense, and the nonsense mutation. We have an example of a codon here, UCA, which codes for serine. Now, based on the position of the base which is being substituted, we may have different consequences. For example, if the base alteration is at the third position, that is, the A here in the UCA is being replaced with U, that is formation of UCU. Now, UCA codes for serine, UCU also codes for serine. Now recall the degeneracy of the genetic code, in which it says that the third nucleotide may not be so important in determining the amino acid which is being coded by this. So therefore, if there is a change in the third nucleotide of a codon, it may result in the incorporation of the same amino acid during the translation process. So that means there may not be any effect in the protein biosynthesis. So such type of mutations are called as silent mutations. So silent mutations occurs when the change in the nucleotide sequence does not result in change in the amino acid which is being incorporated. Now let us consider this second one, where the change is taking place in the first nucleotide sequence over here. So here the U is being replaced with C. Now this results in a different codon CCA, which codes for another amino acid. That is, UCA here is coding serine, whereas CCA is coding for proline. So we have a protein which is being synthesized, where at this point, instead of serine, a proline gets added. So this type of mutations are called as missense. So that is, here because of the change in the nucleotide base, there may be some other amino acid being added over here. So there is a missense. So missense mutations occurs. Now sometimes the change may also result in the generation of a stop codon. Now we can see this in this example. If in the second position, if the C has been replaced with a, now this results in UAA, which is a terminator or stop codon. So such type of mutations are called as nonsense mutations. Now please do not get confused. It's not necessary that the first and second position should always result in missense and nonsense only. It can be, you can have a codon where a change in the first nucleotide may also generate a nonsense, or a change in the second position may also result in a missense mutations. I hope this particular example clears you the different type of mutations. That is, silent mutation occurs when the change in the amino acid does not take place due to degeneracy of the genetic code. Whereas missense mutation says the change may occur such that the amino acid which is being normally has to be coded is being replaced by another amino acid. Whereas nonsense mutation says that now because of this base substitution, there may be a generation of a stop codon or terminator codon. Next, coming to the effects of missense mutations. This image here simply explains the missense mutations. So you can see that a single change has changed the animal. So I have a cat, the C has been replaced with R, it becomes I have a rat. So this is the missense mutations. Now depending on the severity of this missense mutation, that is where the amino acid has been changed, from which amino acid to which amino acid it has been changed, these missense mutations can be acceptable, partially acceptable, or unacceptable. To understand this, let's go to the next slide. The effects of missense mutations can be best understood by studying the structure of hemoglobin. A large number of mutations have been found to be seen in the hemoglobin gene, and this mutations can cause some changes in the structure and functioning of hemoglobin to no change in the functioning of the hemoglobin. So this table I have taken from Harper, which beautifully explains the three concepts, that is acceptable missense mutation, partially acceptable missense mutation, and unacceptable missense mutation. Now coming to the acceptable missense mutation, what it means here is there is a change in the amino acid in the protein structure, but this does not alter the functioning of the protein. Now probably this could be because the change in the amino acid may be at a position in the protein structure which is not critical to the functioning of the protein, or it could be because the changes has taken place in such a way that a similar amino acid with the similar physical properties has been added. This can be best understood with this example, that is adult hemoglobin of the beta chain has got at the 61 position a lysine, whereas in another hemoglobin called as hemoglobin Hikari, which is present in certain Japanese people, now in this position instead of lysine we have aspargine. Whereas at the molecular level, if you see, the changes is in the third nucleotide of the codon. So here AAA or AAG, which codes for lysine, has been altered to AAU or AAC, which codes for asparagine. So that is the acceptable missense mutation, where even though there is a change in the amino acid but has not altered the functioning of the protein. Now coming to the partially acceptable missense mutation, this can be best understood with the example of sickle cell. This is a classical example. So all of you know that in the normal adult hemoglobin at the beta chain in the 6th position, there is a glutamate which is coded by GAA or GAG, whereas in the sickle cell hemoglobin at this position, we have valine which is being coded by GUA or GUG. Now the glutamate is a polar amino acid, whereas valine is a non-polar amino acid. So this has resulted in the alteration in the functioning of the hemoglobin. So here the hemoglobin is not like totally non-functional, but it is partially functional. At high altitudes and in the deoxyhemoglobin state, there is sickling of the RBCs. So that is the partially acceptable missense mutation. Now coming to the final one, that is unacceptable missense mutations. So this occurs when the change in the amino acid has resulted in the loss of functioning of the protein itself. So the best example is the hemoglobin M, in which at the 58th position, the histidine has been replaced with the tyrosine, and this you can see the changes in the codon. So CAU or CAC, which codes for histidine, has been replaced with UAU or UAC. Now this histidine here at this position, 58th position, which is called as the distal histidine, is important for binding of the oxygen to the hemoglobin molecule. Now if this histidine is replaced with tyrosine, now this particular hemoglobin cannot carry the oxygen molecule. So this is an example of unacceptable missense mutations. So that completes your point mutations. So let me revise for point mutations. Point mutations can be based on the base changes. They can be transition or transversion. Based on the effect, we have this silent mutations, missense mutations, and non-missense mutations. Missense mutations may be acceptable missense, partially acceptable missense, or unacceptable missense mutations. Coming to the second type of mutations, that is the frame shift mutations. So frame shift mutations occurs when there is insertion or deletion of one or two nucleotides in the DNA. Now recall in the genetic code that once the genetic code, the reading frame begins, there is no punctuation. That is, there is no comma or any other type of punctuation marks to differentiate the two different codons. So once the reading frame begins, it is continuously read as a triplet codon. So now if there is a insertion or deletion of one or two nucleotides, now the reading machinery, that is the translating machinery, cannot understand that there is something missing or something wrong over here. So therefore, this results in a protein synthesis, which is a totally different protein. To understand this concept, let us take this example. So we have this particular gene here, which showing the different codons and showing the different amino acids which is being coded by this. Okay, so you can see threonine, lysine, arginine, glycine. Now, in this particular gene itself, if there is a additional nucleotide added, that is the insertion of one nucleotide. So what will happen? Now here the translating machinery cannot make out that there is an extra nucleotide over here. So therefore, the reading frame changes. So the first codon ACA is read as it is. So it is threonine. Now instead of AAG, it is taking as GAA, which codes for a different amino acid. And downstream, the entire reading frame changes. So this results in a protein which is totally different from this particular protein. So this is what is the frame shift mutation, which occurs because of a insertion of a single base. Similarly, when there is a deletion, so again in this also you can see that once the first codon has been read, the deletion of A has taken place. So instead of AAG, here the next three are taken, that is AGA, which codes for a different amino acid. So therefore, with this deletion here, again you may have a protein which is synthesized, which is totally different. So frame shift mutation results in production of a totally different protein, that is production of an entire different protein than the original protein. Sometimes it may also result in premature termination of the protein synthesis. That is, generation of a stop codon can take place, which is normally not present at that particular position. Or it can also be that the protein is synthesized in such a way that the stop codon has been changed and there is a read through of this stop codon or the terminator codon. So entirely this frame shift mutations results in a protein which is called as the garbled protein or a totally useless protein. Maybe this particular image explains it best. So you can see that this simple sentence over here, the fat cat sat. So if you just remove one single letter from here, T, you can see that it has resulted in a sentence which is a totally useless. Okay, it does not have any meaning. So similarly, in case of frame shift mutations, because of the single deletion or insertion or even deletion and insertion of two nucleotide bases results in a protein which is synthesized, which is totally non-functional or a garbled protein. Now, what happens if there is addition of or deletion of three nucleotide bases? That is, if it is three nucleotides are being added or three nucleotides are being removed, what will happen? It does not cause frame shift mutations. Rather, it results in a protein which is synthesized, which may have additional an amino acid or there may be an amino acid missing based on whether it is the insertion or deletion. To understand this, let us take this one example here. So you can see that this is the normal mRNA giving rise to this particular protein having these various amino acids. So methionine, alanine, serine, cysteine, lysine. Now, if we remove three nucleotides from this position, that is three nucleotides, that is UGC, then there is a missing cysteine over here. So after methionine, alanine, serine, there is a jump to lysine. So this results in a protein which has got one amino acid lesser because of the deletion. Similarly, if it is an insertion, there may be an additional amino acid. So again, depending on how critical is the amino acid for the functioning of the protein, you may have a protein which may be functional or non-functional. But this does not change the reading frame, so it is not a frame shift type of mutations. There is one more type of mutations called as suppressor mutations. In prokaryotes and lower eukaryotic organisms, abnormally functioning tRNA molecules have been discovered that are themselves the results of mutations. That is, these tRNA molecules occur because of the mutations in their anticodon arms. Now, these tRNA molecules are capable of binding to and decoding the altered codons, thereby they can suppress the effects of mutations. They can suppress the effects of different types of mutations like missense mutations, nonsense mutations, and frame shift mutations. So such tRNA molecules are called as suppressor tRNA molecules. However, these suppressor tRNA molecules are not capable of differentiating the normal codons and the mutations. So therefore, presence of these suppressor tRNA in the microbial cells have found to be decreasing the viability of these microbes. This image here explains very well the different types of mutations, taking a non-medical example. You can see the different types of mutations here. This is normal point mutation with substitution, then frame shift mutations of insertion, deletion. There is one more called as inversion, where the position of the nucleotide has been interchanged. To summarize, mutations are the changes in the structure of the DNA or the nucleotide sequences. So based on the changes, we have the different types of mutations. So it could be point mutations or frame shift mutations. So point mutations is where there is a single change in the nucleotide base and they can be classified as silent mutations, missense mutations, and nonsense mutations. Whereas frame shift mutations occurs when there is a deletion or addition of base or two bases. And based on the substitution of the base, we may also have point mutation classified as transition and transversion. So with that, we come to the end of this particular topic. If you have any doubts, you can always clarify with me via the platforms. Thank you.